Okay, our next video is going to focus on the specific defense or specific resistance, and that is going to focus on immunity. So specific immunity is different than the non-specific resistance that we just talked about because immunity is directed against a particular pathogen. So that's what we mean by specific. Your immune system has memory. So then when it's re-exposed to that same pathogen, the body has a much quicker reaction. Oftentimes there's no noticeable illness because the reaction time is so quick. So our immune system is a large population of cells. They're widely distributed over the whole body. They are able to recognize foreign substances and then act to either neutralize them or destroy them. So you're going to hear some things that sound familiar from the non-specific resistance, but keep in mind this is about specifically directed against a particular pathogen. So before we get into the forms of immunity, this video is just on the overall general aspects of what immunity is, and we'll have a video on each of these types. So humoral is antibody-mediated immunity, and whereas cellular is cell-mediated immunity. So cellular means that the cells directly attack and destroy the foreign cells, or humoral is they're not directly destroying the pathogen. It's kind of an indirect using antibodies. Again, we'll have a separate video for each of these types. But before we can talk about the separate types, we need to kind of classify. There's 4 types of immunity. The immunity can be classified into. And so let's go through those. Natural active immunity is when you are producing your own antibodies or T cells because of an infection or a previous exposure. Natural exposure. That's just natural active immunity, and that's what we're going to focus on. But there's also some other types. Artificial active immunity is when you are producing your own antibodies as a result of a vaccination. So you weren't exactly exposed to the disease, but you were exposed to an artificial version. So a vaccine is dead or weakened pathogens that stimulate this immune response without actually having the disease. So that's why it's called artificial active immunity. Booster shots are periodic immunizations to help maintain that memory. And then we also have 2 types of passive immunity. So natural passive immunity is temporary immunity. It can be from antibodies produced by another person. The only example of this that we have is a fetus acquiring antibodies from the mother through the placenta or through milk if they are breastfeeding. And then finally, artificial passive immunity. This is temporary immunity from the injection of a serum, which is antibodies from another person or animal. And an example here is if you get a venom for a venomous snake bite or rabies, tetanus, and so most of those treatments are coming from other animals. But we're going to focus on the natural active immunity. So before we get too far into that, we need to define a few terms. Antigen gets used a lot, so let's define that. Antigen is any molecule that triggers an immune response. Okay, so that's pretty broad. They're complex molecules that are unique to an individual and large. So that's what an antigen is. You've talked about that a little bit when we were working on blood types. But there is on that antigen, there is a region of that molecule that specifically stimulates the immune response. So there's just one little part of it, or region, that is what actually triggers all these responses that we're going to be focusing on. Okay, there are some molecules that are too small to be considered antigens. They're heptins, but they do combine with a host macromolecule. That's a large molecule, and so they are still able to help the body recognize some foreign molecules. So cosmetics, drugs, detergents, poison ivy, animal dander, and so with subsequent exposure, then there's going to be a response triggered. If you're ever having trouble with these, I've noticed it depends on which computer I'm using, but just a side note, if you're ever having trouble with these, you can always just look them up separately on YouTube and watch them, and I would encourage you to do that. All of the animations that are embedded within these PowerPoints. Okay, so let's talk about the cells of the immune system that are focused on this immunity. We've already talked about those NK cells. So in this section, we're going to talk about T cells and B cells. Those are the cells that we're going to focus on as far as specific resistance. Again, we've already said this earlier, but these lymphocytes are going to be more concentrated in the lymphatic organs and in the skin and in mucous membranes, because those are the places that those foreign particles are going to be more concentrated. So T cells are created in the red bone marrow and then released into the blood. They're still stem cells, and then they're going to move to the thymus. So in the thymus, they're going to be stimulated, and they get these surface antigen receptors, which is exactly what it sounds like. So it's a receptor on their surface for antigens. And once the receptors are in place, the T cells are now able to recognize antigens. Okay, so once they are mature T cells, this immunocompetent means that they're able to detect antigens, and it's going to be important what antigen they are detecting. How they test them is these RE cells, this reticuloendothelial cells in the thymus. They're going to test those T cells by presenting self-antigens. That means cells that have your own antigen on the outside, and that's how they're going to test the T cells before they release them. But the T cells could possibly fail the test. In fact, many of them do. Maybe they don't recognize the correct antigen. If they don't recognize their own antigen, then they're going to have trouble recognizing a foreign antigen. Or if they react to the self-antigen, that's also a problem, because then they would be attacking their own tissues. So T cells that fail the test are not going to move out of the thymus, and that's called negative selection. So T cells that fail the test it's called negative selection. They must be eliminated. So they're either die right there in the thymus, or they remain alive, but they are unresponsive. So they are not going to be activated, if you want to think about it that way. Only 2% of the T cells that reach the thymus actually leave. They actually graduate and leave. They're able to assess those antigens and be able to recognize them. And so then they move to the medulla of the thymus to undergo what's called positive selection. So the 2% that have made it, they are actually going to multiply and form clones of identical T cells programmed to respond to a specific antigen. Remember, this is specific for a certain pathogen. So if there's a certain pathogen, then we want a specific antigen to respond to that. I'm going to actually jump a little bit past this part about B cells, except that I will talk about B cells are very similar to T cells, but they are synthesize what's called those antigen surface receptors. And so they are going to act as antigen presenting cells. So let's go back for just a second. So because they are able to make those antigen surface receptors, then they are also going to make clones of themselves. But then we're going to see in this next section that they are going to be helpful in finding those specific foreign antigens. So T cells can't find the foreign antigens on their own, and so B cells are going to help with acting as an antigen presenting cells. I know all these long names. An APC. And so they basically help the T cells identify the certain antigens that are foreign. So not only do they identify the antigens that are foreign, but they also have self-antigens. So they are going to identify all cells for you, but then they also are going to be able to, if they know it's not a self-antigen, then it must be a foreign one. So what happens with these APCs, or these antigen presenting cells, they have a little bit of a process. They're going to come encounter the antigen. They're going to basically internalize it by endocytosis. We're going to see a picture here in just a minute, which basically means they're going to engulf it and then digest it into smaller pieces, and then display those pieces on the outside of their cells. So I think this is hilarious, but this is what it looks like. Your book calls it little hot dog buns. So let's kind of take a picture or look at this picture. So here's an example of an antigen presenting cell. It's going to come in contact with the antigen in number one, bring it inside by endocytosis, break it apart with enzymes, and then kind of put that little fragment out on the outside of where that MHC protein is. And so that is going to be displayed on the surface of the macrophage. And so then it's presenting. It's basically wandering around. And so those T cells inspect them for those displayed antigens. So they go up to the APC and see what they have displayed. And if the APC has a self-antigen display, then the T cell doesn't worry about it. It already knows that that's my self-antigen. I'm comfortable with that. But if it's a non-self-antigen, then the T cell is initiating an immune attack. So if it's a foreign antigen, a non-self-antigen, and the T cell notices it, and then that's when this immune attack is going to start.